Hello to whoever is watching this video. Well, here is a video I have made on one of the topics of Big Idea 3 in AP Biology. So, as you can see ahead of you, this is an outline of what we're going to go over in this topic discussion in outline 3. Of an outline of Big Idea 3, which is the transfer of information from genes to proteins, which are used to be expressed for specific traits and phenotypes. So this is a very important process and the reason why we express proteins that we do. So this is how it goes, basically. So for the outline right here, it explains what we're going to go over in a timely fashion. So first is DNA structure, and then the aspects of replication. Just hit them softly, since we're not going over the specific topics of that. Then transcription, translation, and then the aftermath of what happens to those specific proteins that are synthesized from this process. So let's first start off with this DNA structure. Well, as you can see here, this is the DNA structure of nitrogenous base pairs and such. So, what does DNA stand for? Which stands for deoxyribose nucleic acid. So, what's inside DNA is first a deoxyribose, which is an oxidized ribose group, which is kind of like a sugar. And then then there's 4 nitrogenous bases, which is as you can see, this is color coded up here. It is thymine, adenine, guanine, and cytosine. So those are specific to DNA. And then also a phosphate group. So what the phosphate and the deoxyribose is used for in DNA, in this double helix structure, DNA is always in double helix structure. What it is that it's used for the backbone. So as you can see here, in the backbone, what's in it is the deoxyribose and then the phosphate. So, but these are the backbone of DNA. And then what actually is in the middle of it are the nitrogenous bases. So as you can see below, I wrote down that the nitrogenous bases, thymine and guanine, are specific bases called pyrimidines. So pyrimidines are actually smaller than purines, which are another type of nitrogenous bases. So these have to be able to fit within the DNA, double helix. So thymine is a pyrimidine that's always going to bond to adenine, which is a purine. So these will always be in equal amounts in the DNA because one is always complementary to the other, as you can see here, there, and further on. And then same for guanine and cytosine. They're always going to be in the same amount because they're complementary to each other. So for example, if I say that I'd say 60% of the cell, how about 30% of the DNA is thymine. Therefore, find out what's the rest of it. So we can infer that 30% is also adenine because these are always complementary to each other. And then also we can say that guanine is 20% and then cytosine is 20% because 60% is adenine and thymine. So therefore the other 40% is guanine and cytosine, and then they're both split up because they're both complementary to each other. So another important fact is that these are actually bonded by hydrogen bonds, which are in the inside of the DNA, the DNA structure. So since it's always in a double helix. All right. So I'm gonna touch lightly on the the replication of DNA. So as you see here, this is something called the semi-conservative model, which I'm going to actually model for you. So semi-conservative model, what actually states is that the parent strand remains with the newly synthesized strands when DNA is replicating. What you'll see in a few moments how it actually works is basically that when DNA is synthesized, so say these are old strands right here, when it's synthesized into 2 new strands, the older strand, I'll just shade it in a little darker, and then you have a new strand. It's pretty light right here. But the old strand is always going to remain with a new strand because it's always synthesizing a new strand. And then the old strand is going to be whatsoever. You'll learn. It's the same as it goes. So here's just a simple model to show you how it goes, basically. So pretend these are nitrogen bases. You can say this is guanine, this is cytosine, this is adenine, this is thymine. Alright, so what is going on here is that you have a DNA. We're going to act like, this is like DNA not in a double helix structure. And then you see this is the phosphate sugar backbone and then it's nitrogen bases. So when we get to replication of DNA, there's actually some main players that are involved in it. So first we have the main players. First of all, we have, here they are. First, we have, let's start off with helicase, topoisomerase, and single-strand binding proteins. Alright, I'll just put this right here so you can keep reading. But what this is that since they're main players, this is our helicase. So what helicase's job actually is to split up DNA from its chain, basically unwind it. So if helicase, our little enzyme, is going to run through the DNA and split it up. Yeah, just split it up from the replication fork. So DNA is usually long, so it'll split it up into a kind of like a bubbled area. So what's going to occur next is that we need something called primase. So right here we have primase. So the job of primase is very specific. It's actually to create primers on the DNA strand. So it creates primers. So bam, then it creates a primer right here. So what's so important about the primer is that the DNA polymerase, this right here, DNA polymerase cannot be initiated without without a primer on the DNA itself. So once the primer's here, the DNA polymerase can initiate to the DNA, and then it can start synthesizing from the strand. Therefore, since the top strand is 5' to 3', yes, I'm talking about this up here, then it'll synthesize DNA in 3' to 5'. Alright, so synthesize it backwards. Alright. So, next, and then you'll have some others, such as DNA ligase. So for this strand down here, it mostly consists of something called Okazaki fragments, which are small fragments of DNA, DNA parts. So DNA ligase is responsible for binding them together and making sure that they're just bound together. Alright, next, let's get into the main topics of this. We'll move by. First, we have translation. Oh no, I'm so sorry. Transcription. Minor setback. So what does transcription sound like? It's mostly sounds like transcript. So what's actually going on here is that a transcript is being produced from DNA. So this is a messenger transcript. In our case, it is mRNA. So first, in initiation, something called RNA polymerase, going to be right here, is going to bind to a DNA at the promoter sequence, and then it's going to initiate DNA, initiate translation, and then start translating. So what's actually the difference between this RNA polymerase and then DNA polymerase is that RNA polymerase does not need primers to be initiated. Therefore, it kind of rules out DNA polymerase. So, in elongation, this RNA polymerase is going to continue moving down the DNA strands, synthesizing just off of one strand, basically. So if it's synthesizing off the 3' to 5' strand, then the transcript is going to be synthesized as 5' to 3'. So, alright, it's going to keep moving along, and then during the last step of termination, it's going to read what's called a termination sequence. So up here, I kind of wrote it down. So what a terminated sequence is a specific sequence that the RNA polymerase is going to read. And once it reads it, it's going to know to stop translation and jump off of the DNA strand, and then well, basically not jump off, but unbind from it, and then then cleave the mRNA strand, and then we have a mRNA transcript. So it's just some extra important facts is that RNA transcript, the mRNA transcript is not going to contain thymine. So instead of the nitrogenous bases of including thymine, it's going to be guanine, cytosine, and then it's going to be adenine and uracil. So it's going to be the U instead of T. That's an important fact about RNA, that it does not have thymine. And next we have what type of RNA is used in this process. As I mentioned before, it's mRNA because it's the messenger. Lastly, translation. So translation is, what does it sound like? Sounds like translating. So what actually going on here is translating probably into a different language. So it's simply going from the language of genetic code and nitrogenous bases to polypeptides and codons. Alright, so what's going to first occur in initiation is that the mRNA is going to move out into the cytosol. So once it's in the cytosol and other stuff's been done to it, the small subunit of the ribosome is going to bind to the bottom of it. And then the large subunit is going to come along and bind to the top. Therefore, RNA is already in the middle and it's going to start moving. But it's going to start moving during elongation. So what's specific here is that there's something here. What these things right here are called their tRNA. It's kind of important because tRNA, they actually carry polypeptides with them. So these polypeptides, what they actually have, these tRNA, at one end they have a polypeptide, at the other end they have something called anticodons. So during translation, these tRNAs, they read the mRNA in triplet pairs of nitrogenous bases. So they'll read each in a pair of 3. So with the start codon here, that's going to start off the initiation of it and start off the first nitrogenous. That's going to start off the first polypeptide to be synthesized. So for each, since there's only 64 there's 64 different combinations of codons, yet only 24 there's only 24 polypeptides that can be produced from it. So that's because the flexibility of the third codon, but it's going into a whole nother topic. So what's going on here is that first, during the elongation of it, these anticodons, which are basically the opposite of the regular codons and their bases, are going to specifically bind to the mRNA. And once they're bonded at a specific, I'd say they have to be very specific for the specific codon. So once they bind to it, what's going to occur is that in the long process of elongation, once this is in the A site, you see here is the EPA site. So for the EPA side, once this is going to first go to the APE side, that's when it's binding to the anticodons, binding to the codons of the mRNA. So once it binds here, this the codon that's in the P side already, the peptide side, what it's going to do, it's going to give the whole chain to the next codon. But this codon is actually also moving into the P site, and the codon that was in the tRNA